This is Chapter 10 on photosynthesis. Photosynthesis is the use of light to make chemical energy, and that chemical energy is going to get stored either as sugars or other organic molecules. We have organisms that are able to produce their own food, and other organisms are relying on food to be produced by a different organism. The autotrophs are the self-feeders. They're going to produce organic molecules from either carbon dioxide or other inorganic raw materials from the environment. They're not dependent on other living beings, so they're considered producers. The heterotrophs are going to obtain their organic materials by either consuming compounds produced by other organisms or the organisms themselves. They're considered the consumers. A photoautotroph is an organism that specifically uses light energy to drive the synthesis of the organic molecules from carbon dioxide. So photosynthesis is going to be where we convert light to chemical energy. So you'll notice there is some areas highlighted in green here on the notes. You're going to want to make sure you go through your notes and update these. Some of the versions of the notes have had these corrections where some of the terms, the lines got mixed up, and some of the versions of the notes do not have it. So anything that's highlighted on here, please make sure you double check your notes. So the chloroplast is the site of photosynthesis in plants, and within the chloroplast you're going to have chlorophyll, which is a dense fluid. It's actually the pigment that will absorb the light energy. The mesophyll is the part of the leaf where the chloroplasts are found. It's in the interior of the leaf. The roots are going to be responsible for absorbing water and delivering it to the leaves through the veins. So it's only the green parts of the plant that are going to do photosynthesis. The stomata are microscopic pores where carbon dioxide is able to enter the leaf and oxygen exits. And then the thylakoids are a system of interconnected membranous sacs. It's going to segregate the stroma and chlorophyll in the thylakoid membranes. The stacks of thylakoids are known as grana or granum. So this picture here is showing a cross-section of a leaf. So you have the chloroplasts, the veins. Here you have the stomata with the carbon dioxide going in and the oxygen going out. Within the chloroplast we have the thylakoid sacs that are stacked in these granum. And then you can see the stroma is labeled here around the outside. So when we look at the reaction for photosynthesis, all of the atoms are accounted for. We start with 6 carbon dioxides, 12 water molecules, and react it with light energy to make 1 glucose, 6 oxygen, and 6 water. So this diagram here just shows where you can see all of the carbons are accounted for, all of the hydrogens are accounted for, all of the oxygens are accounted for, so we can be sure that we've observed the idea of conservation of matter or energy. So in the equation we use glucose to simplify it, but what's actually going to be produced is going to be a 3-carbon sugar, and we will make multiple of those 3-carbon sugars that can then be used to make glucose. One of the things that can be helpful in understanding photosynthesis is to go back and look at cellular respiration and kind of see how the processes are similar and how they are different, and they kind of go in the reverse direction. So when we look at the straightforward equation here, we have 6 carbon dioxides plus 6 waters plus light energy to make glucose, 1 glucose and 6 oxygens. If we simplify the equation here, you can see carbon dioxide plus water is going to give you a sugar. In the parentheses here, this is going to be, all sugars would be multiples of CH2O. So if it was a triose, you would have 3 CH2Os. A pentose would be 5 CH2Os. And with glucose, there's 6 of them. So this is an overview of the process of photosynthesis here in this diagram. It's occurring in the chloroplast. You can see the light comes in and gets absorbed along with water. You're going to have the light reactions. They're called the light reactions because they need light to occur. You're going to have oxygen released out at that point. Here you're going to create some ATP as a source of energy to drive the Calvin cycle, and you're going to produce NADPH as a source of electrons for your reduction and oxidation processes. The Calvin cycle is over here, where you're going to take in carbon dioxide. You're going to take in multiple carbon dioxides and essentially use the ATP to glue them together and then release the sugars. What remains behind has to be regenerated so you can continue the cycle on. So in the first stage you have the splitting of water. Oxygen is going to be given off by the plants and is going to come from the chloroplast when they split the water into hydrogen and oxygen. So C.B. Van Niel actually proposed that all photosynthetic organisms require hydrogen, but the source varies. So we can look and see the same general reaction here. In plants, this general reaction has the two hydrogens attached to oxygen. In the sulfur bacteria, these two hydrogens are attached to a sulfur. So we have this general equation here where you have the hydrogens attached to this X, and then you have the products released over here. If it's the sulfur bacteria, it's two sulfurs released. In plants, it's going to be the oxygen gas released, and just the general equation is two X's. So there are other microorganisms that are going to use this sulfur bacteria for one, and others will use the general equation with other elements as a source of their hydrogen. So photosynthesis is a redox process, so there will be electrons being moved. In respiration, energy gets released from the sugar when the electrons associated with the hydrogen are transported by carriers to oxygen. In photosynthesis, electrons are going to increase the potential energy as they move from water to sugar, and that process is going to require energy from light. So photosynthesis has two stages. The first stage is the light reactions or light-dependent reactions. Here is where we're going to convert the solar energy into chemical energy. NADP+, or nicotinamide adenine dinucleotide phosphate, is an electron acceptor. So solar power will reduce the NADP+ to NADPH. With photophosphorylation, you use the light reactions to generate ATP, and you use chemiosmosis to power the addition of phosphate to ATP. In the Calvin cycle, here is where you're going to take in the carbon dioxides from the air and attach them together to make the organic molecules in the chloroplast. It will then be reduced to a carbohydrate by adding electrons. So it gets that reducing power from NADPH, and the chemical energy comes from ATP. That initial incorporation of carbon into the organic compounds is carbon fixation. So in photosynthesis in the plants, we're incorporating carbon dioxide. So our light reactions are going to convert solar energy to chemical energy in the forms of ATP and NADPH. So this is something you'll want to update in your notes here as well. So with sunlight, it is a wave, and when we look at waves, they're part of the electromagnetic spectrum. We look at the wavelength, which is the distance between the crests of the waves. Your electromagnetic spectrum is going to be a range of wavelengths from less than a nanometer, which is going to include your gamma rays, to more than a kilometer, which will be radio waves. Let's see if I can get this to center on the screen here. So you can see the gamma rays on the left side, the radio waves, In here, this little sliver is expanded out. This is going to be your visible light. This is the only part that you can see. With the wavelengths, they are shorter on the left side here and longer over here. The shorter wavelengths will have higher energy. The longer wavelengths will have lower energy. The way I remember it is I just make an association where you've got shorter wavelengths like smaller dogs, they're higher energy. I think of the long wavelengths like big dogs, your big old dog that slowly walks around, that's low energy. So it works for me to remember the difference. You're welcome to use something else if it helps you to remember the difference. So the higher energy waves do have more potential to cause more damage to human tissue. So down here we have to limit our exposure to, actually UV light has enough energy that it can cause damage, but we limit the exposure to X-rays and gamma rays. If you're around these types of waves long enough, they could actually cause damage as well. So visible light is just the segment that's from about 380 nanometers to 750 nanometers. It's important to life, and light behaves as a wave, but it also behaves as a particle, so it does have a bit of a dual nature to it. The photons are the discrete particles of light. So they are not tangible, but they actually do have a fixed quantity of energy, and the amount of energy is inversely related to the wavelength of the light. So the shorter the wavelength of light, the greater the energy of each photon. So the photosynthetic pigments are your light receptors. Pigments are just substances that absorb visible light. So different pigments are going to absorb different wavelengths, and the absorbed wavelengths are going to disappear. What we actually see is the part that is not absorbed. So when you see something green, what you're seeing is the green wavelengths being reflected back. Spectrophotometer is an instrument that you can use to measure the ability of a pigment to absorb various wavelengths of light. And then the absorption spectrum can be plotted with the absorption versus the wavelength in the different pigments. So the chloroplasts have three different pigments. Chlorophyll A, which is going to participate directly in the light reactions. Chlorophyll B, which is an accessory pigment, and the carotenoids are a group of accessory pigments. So your action spectrum of these profiles the relative effectiveness of different wavelengths in driving a process. So for chlorophyll A, the violet, blue, and red light works the best. Absorption of chlorophyll A alone underestimates the effectiveness of certain wavelengths in photosynthesis because of the accessory pigments. So your chlorophyll A is the blue-green, chlorophyll B is the olive-green, and the carotenoids are going to be your shades of yellow and orange. They help broaden the photosynthetic spectrum. So here we can see the photosynthetic spectrum. So when you look at leaves, in the spring and the summer when they're green, they still do have the carotenoids. You're just not seeing them because of the chlorophylls being more robust and healthier. In the fall, when the chlorophylls start to dry up, that's when you see the carotenoids that were always there anyway. Another important role of some of the carotenoids is photoprotection. It helps to absorb or dissipate excessive light that would damage the chlorophyll or interact with the oxygen. So it is actually possible to sunburn a plant. So when we excite chlorophyll with light, you're going to have a molecule absorb a photon of light. So right here we can see the photon coming into the chlorophyll. And when that happens, one of the electrons gets elevated to an orbital with more potential energy. So down at the bottom here is the ground state when it's in its normal orbital. Up here we refer to this as the excited state. So the energy is going to eventually have to come back down. This energy that is in here still has to be accounted for. So it can be released as heat. Another way that it can be released is as a photon and lead to fluorescence. So the only photons that are absorbed are those whose energy exactly is equal to the energy difference between the ground and excited state. So this will vary from one type of molecule to another, and this is why you need to have different pigments from different absorption spectrums. The excited state is unstable, so the electron is going to drop back down and release that energy. So some will release it as heat, some will release it as heat and emit light. The afterglow or fluorescence is what you see when it emits light as the electron drops back down. So your photosystems are going to be your reaction center complex associated with your light harvesting complex. So in your photosystem, you have a protein complex called the reaction center. And that complex is surrounded by several light harvesting complexes. So here at the top of this picture, you can see you've got your reaction center complex, and then you've got your light harvesting complexes around it. Your primary electron acceptor is going to be the molecule that's capable of accepting the electrons, and it becomes reduced in the reaction center complex. So up here, this represents your primary electron acceptor. So chlorophyll A uses energy to boost an electron, and it transfers it to a primary electron acceptor. So here you've got chlorophyll A. You've had the energy come in, the energy get transferred. Here you have the electron get elevated. So we have two different photosystems. Your photosystems were named in the order they were discovered, not in the order that they are found. So photosystem 2, or PS2, functions first. It's the reaction center chlorophyll A, and it's considered P680 because it absorbs wavelengths at 680 nanometers. Photosystem 1, or PS1, has chlorophyll A with a P700. It's in the far red spectrum. And it's going to have a slight difference in the absorption spectrum from photosystem 1 to photosystem 2. So here you can see we've got photosystem 2 that comes first. The energy from the light comes in and is going to be passed along. The electron gets excited to the primary electron acceptor. It's going to get passed down this electron transport chain. You're going to produce some ATP in the process, and it's going to get elevated again. There'll be a little bit more light that contributes to this, to another primary electron acceptor, and get passed down a second electron transport chain. So we refer to this as linear electron flow because the electrons are going to flow through the photosystems and other components of the thylakoid membrane during the reactions. So the electron starts at one end and it ends up over here. What that means is you're going to constantly need a source of electrons here to keep these reactions going. So this is just a verbal description here. I'm more concerned in you having just the big picture idea rather than knowing where you have the different cytochromes, the plastoquinone, plastocyanin. As long as you know the big picture that it's going to excite in photosystem 2, then excite in photosystem 1, passing to the primary electron acceptors, and which one uses P680 and which one uses P700. That's going to be adequate. So with cyclic electron flow, it uses just photosystem 1, but not photosystem 2. So here the electron is going to cycle back to the original reaction center, and so you don't need a continual new source of electrons. So the electron cycles back to the cytochrome complex in the P700. There's no NADPH and no release of oxygen, but it will generate ATP. So this is done by some of the photosynthetic bacteria, also with the purple cellular bacteria. So in this picture here, you can see what is in color is what you have in the cyclic system versus in black and white, the additional parts that you have with the linear electron flow. So here you can see the electrons are going to flow back to the original reaction center complex. They are not getting passed down this chain to create the NADPH. So you can see there's an electron transport chain just like you have in the mitochondria here. So the chemiosmosis that occurs in the chloroplasts is very much similar to the one that occurs in the mitochondria. Both are going to generate ATP through chemiosmosis. It will have some of the same electron carriers, including the cytochromes. The ATP synthase complex at the end is similar. The source of electrons in the mitochondria comes from organic molecules. The source is water from the chloroplasts. Mitochondria uses the chemiosmosis to transfer your chemical energy from food to ATP. Chloroplasts are going to use it to transform light energy to the ATP. In the mitochondria, the inner membrane pumps the hydrogen from the matrix to the inner membrane space. In the chloroplasts, it's going to pump the hydrogen from the stroma into the thylakoid space. So here you can see similar structure of the mitochondria and the chloroplast. And you can see here in this picture that you've got this ATP synthase at the end, so that when you have the hydrogens that are all on the one side of the membrane and they come across through the ATP synthase, they will help to phosphorylate ADP to make ATP. So similar process in both the mitochondria and the chloroplast. So this just shows here that process of the light coming in, and instead of showing it just in a big circle, it's showing it actually embedded here in the membrane of the thylakoid space with your linear electron flow, sending the electrons down this electron transport chain. Here with photosystem 1, a little bit more excitement, moving it up to the electron acceptor and it getting passed down again. And then you will send that ATP to the Calvin cycle as well as the NADPH. So in the Calvin cycle, you're going to use the ATP and NADPH to convert carbon dioxide to sugar. The Calvin cycle is actually similar to the citric acid cycle in regenerating its starting material. So the citric acid cycle, though, is catabolic. You're breaking things down. The Calvin cycle is going to be anabolic. You're going to be building something. So in the Calvin cycle, it's going to produce the sugar glyceraldehyde 3-phosphate. So the cycle has to take place 3 times and fix 3 carbon dioxides into glyceraldehyde 3-phosphate. So fixing 3 carbon dioxides means you're going to incorporate 3 carbons. So here at this point, phase 1 of carbon fixation is where you're going to be bringing in the carbon dioxide. It's going to come in and attach to the rubisco. I'm not so concerned about you knowing the names of the intermediates here, but if you know the first phase, carbon fixation, that it brings in the carbon dioxide. So it gets added on. You've got the 3 carbon sugar here. It's going to have energy being supplied by ATP. You're going to have a source of electrons for reduction. So in phase 2, we have the reduction. The electrons are being put in. And then you're going to pull off glucose or glyceraldehyde 3-phosphate here that will ultimately get turned into your sugar. In phase 3, we have the regeneration. Here we're going to go through and we're going to reorganize the molecules again so that we can regenerate our carbon dioxide acceptor, the RUBP. So this area here gives details on what is actually going in on the process. I'm more concerned about you having the big picture idea rather than knowing all the intermediates. The information is here if it helps you to be able to look and see, okay, now I understand why I'm summarizing this, but I am not going to expect you to produce that level of detail on the test. So we do have some alternative mechanisms of carbon fixation. So arid climates can create a bit of a challenge for plants. The stomata are going to lose water as well as allowing the carbon dioxide in. If you close it, you're not going to have carbon dioxide come in, so the plant has to either close the stomata on hot, dry days, which is going to decrease its photosynthetic yield, or it will become dehydrated. So when you decrease the photosynthetic yield, your carbon dioxide decrease in the air spaces and oxygen released begins to increase. So it creates a wasteful process of photorespiration. So instead of having the carbon dioxide to incorporate, you're not going to be actually producing the sugars. So photorespiration occurs in the C3 plants. Your first product of carbon fixation is a 3-carbon compound, 3-phosphoglycerate. These end up producing less sugar because they have decreased carbon dioxide. You can't make sugar if you're not taking in the carbons to add on to the skeletons. Some examples of plants that are C3 plants: rice, wheat, soybeans. The photorespiration occurs in the light. The rubisco binds the oxygen in place of carbon dioxide. So the product splits and you have 2 carbon compounds that will consume oxygen and produce carbon dioxide. So these end up consuming ATP instead of generating it. So your C4 plants have created an adaptation. They're going to practice the Calvin cycle with an alternate mode of carbon fixation that forms a 4-carbon product. Examples of these plants would be sugarcane and corn. There's several thousand species that use this. So here they've got bundle sheath cells. These bundle sheath cells are arranged in tight packed sheets around the veins of the leaf. So you can see them right here. And then the mesophyll cells are loosely arranged between the bundle sheath and the leaf surface. So the Calvin cycle is confined to the bundle sheath cells, but it's preceded by incorporating carbon dioxide into organic compounds in the mesophyll cells. So what happens is you are separating where part of these reactions are done so that you can reduce the amount of water that gets lost. So again, there is a more detailed explanation of it here. My concern is that you've got the C3 plant that's going to become less efficient. You'll have the C4 plant that's going to separate where it actually has the reactions occur. So to do this extra ATP, the bundle sheath cells only have photosystem 1 and do cyclic electron flow. The mesophyll cells are going to pump the carbon dioxide in to concentrate it for the rubisco. So it minimizes the photorespiration and enhances sugar production. So we have one more adaptation, and these are the CAM plants, which stands for crassulacean acid metabolism. This is going to include a lot of the succulents, cacti, pineapples, etc. These are going to open their stomata at night and then close them during the day. So it's going to help decrease the water loss during the day, but there will be no carbon dioxide that enters during the day. So at night they take up the carbon dioxide and incorporate it into a variety of organic acids. So the mesophyll cells will store those organic acids in vacuoles till the morning when the light comes in, and then you can do the light reactions to supply the ATP and NADPH for the Calvin cycle. So this is just showing here a comparison C4 plants who have spatial separation of the steps, with the CAM plants who have temporal or time separation of the steps. So photosynthesis is important. It's only the green cells of the autotrophic parts of the plants. The rest of the plant has to rely on organic molecules being exported from the leaves via the veins. So if you pull all the leaves off of a plant in the summer, it's not going to be able to generate any sugars. Some will form glucose for other anabolic pathways. Some form sucrose. The glucose gets linked into cellulose, which is the most abundant organic molecule in the plant. Some of them will stockpile the extra sugar as starch. So this tends to happen in the ground with some of them, like the tubers. Your potatoes, beets are examples of plants that do that. So this is the end of the lecture on photosynthesis. At some point in the near future, I would encourage you to go back and make the comparison between cellular respiration and photosynthesis. It will just help to clarify some of this in your mind.